Okay, I think let's get started in the interest of time. And thanks everyone for joining. And today we're really happy to have Katie. Katie's a new faculty in McMaster University in Canada, where she does a lot of cancer genomics work that you will hear about. And she has previously trained under Paul Botros, who was in Canada, who moved to UCLA, where I got to know her. She did a lot of brilliant work with us collaboratively as well. And then she went on to do her postdoc in Stanford, which amounts to this work today. So I think with that, Katie, look forward to your presentation.
Great. Thanks so much, Quan. I appreciate that kind introduction. I'll just share my screen and hopefully confirms you see it in the correct mode. Great. Awesome. Thanks so much. It's a pleasure to present and be here today and share this story. So as Quan mentioned, I'm now an assistant professor at McMaster University, which is in Hamilton, Ontario. But this work was done as part of my postdoc at Stanford with Christina Curtis. And it's really diving into an underappreciated role of the germline genome and its involvement in immunoediting, more than maybe thought before. So we're going to take a real big step back and just ask, what causes cancer? Well, we know that cancer is caused by genomic or epigenomic aberrations that can lead to dysregulated cellular processes that we often refer to as the hallmarks of cancer. And the causes of these genomic and epigenomic aberrations can be broadly stratified into these three categories. We have environmental, referring to UV radiation, tobacco, exogenous compounds that induce DNA damage. We have genetic, referring to inherited polymorphisms that may predispose an individual to a particular cancer. And then finally, we have stochastic, which here I'm referring to replication-associated mutations or mutations that just seemingly occur by chance. And these three factors work together to form tumors whose somatic mutation landscape really varies quite drastically from tumor to tumor. So this might be a plot that some of you are familiar with. This is data from the Cancer Genome Atlas, where each point represents a tumor ordered along the x-axis by cancer type, and the y-axis shows the number of somatic mutations per exome. And of course, there's a lot of variability across cancer types, but really what I want to draw your attention to is the span of variability within a single cancer type. So the somatic mutation burden within a cancer type can vary by orders of magnitude. But if we take a step back, we know that we're all born with inherited variability. In fact, our genomes can vary at millions of polymorphic sites. And so this is data from the 1,000 Genomes Project, and I kind of like it because the plot nicely mirrors the TCGA somatic mutation plot on the previous slide, where each dot represents an individual, and the y-axis, this time instead of somatic mutations, showing now the number of inherited or germline variants per genome along the y-axis. You can see individuals of European ancestry here in blue have more than 4 million variant sites per genome. And this increases to about 5 million when we consider individuals of African ancestry. So this begs the question, does this inherited variation actually influence the somatic variability we observe in the tumor genome? And while this is if you think about it, these somatic mutations occur in the context of our inherited genome. And while this may not be completely deterministic, perhaps this inherited variation may bound the likelihood of specific stochastic mutations. And if this is true, how does this happen? So one thing we do know is that the germline genome can modulate immune responses. So this is again work from the Cancer Genome Atlas, who showed that T cell infiltration and cytotoxicity, as well as interferon signaling, is estimated to be about 15 to 20% heritable, meaning 15 to 20% of the variability in these immune pathways can be attributed to germline genetics. And this is important when thinking about somatic mutations because increasing lines of evidence suggests that avoidance of the adaptive immune response is a strong determinant of which somatic mutations can persist in a tumor. So this might be review for some, but somatic mutations that are occurring in protein coding regions of the genome can generate these neopeptides, or these novel 9 amino acid peptides, a subset of which can be presented on the tumor cell surface via the MHC class I complex. And together these form an epitope, a subset of cytotoxic T cells with T cell receptors that can identify this epitope can actually trigger tumor cell death via release of cytotoxic granules. Now, these immunogenic epitopes, so this MHC class I and peptide combination, are largely thought to be derived from somatically altered peptides or abnormally expressed protein sequences. And this is because germline variation or inherited variation should be considered self. And we have tolerance mechanisms that should dampen immune response to self to avoid autoimmunity. But increasingly, we're learning that there are specific circumstances that can facilitate escape from tolerance. There's actually similar numbers of peripheral cytotoxic T cells that can recognize self versus those that can recognize foreign antigens. They're just held in a suppressed state by a peripheral tolerance mechanism. But like I said, there are circumstances that can facilitate escape from this tolerance. And so I've listed a few here, but these include weak epitope binding affinity, post-translational modifications that are restricted to peripheral tissues, and changes to peripheral tolerance. And so this really got us thinking, could we more systematically investigate circumstances in which our inherited variants may actually trigger an immune response? And could this be a mechanism by which our germline genome influences the somatic mutational landscape? And so this leads me to the hypothesis of this work, which was that the germline-derived epitope burden in an oncogene may select against oncogene amplification. What do I mean by that? Okay, we're going to walk through this toy example. Here we have gene X. Gene X is set to represent a recurrently amplified oncogene in cancer. We're going to be talking about breast cancer, so we can imagine gene X is HER2, which we know is recurrently amplified in a subset of breast tumors. Here we have individual A. Individual A has a germline sequence of gene X and a specific subset of HLA genotypes, such that let's pretend there are 2 peptides that can be derived and presented on the tumor cell surface, specifically from gene X in this individual. If this individual develops cancer and the tumor cells develop a somatic amplification of gene X, such that they now have 3 copies, each individual copy produced 2 epitopes. We now have 3 copies. 2 times 3 equals 6. We contrast this now to individual B, who has a germline polymorphism in gene X, as represented by this little orange A, and a different subset of MHC class I alleles. Let's pretend for this individual, there's actually 3 peptides that can be derived from gene X and presented on the tumor cell surface. So if this individual develops a somatic amplification of gene X, 3 copies, 3 epitopes per copy, 9 resulting epitopes. And finally, you can probably see where I'm going with this, but we have individual C, who has a different germline polymorphism, this time a G, as represented by this little blue G, a different subset of MHC class I alleles. Perhaps there's only 1 peptide that can be derived from gene X in this individual and presented. If this individual develops 3 copies of gene X, 1 peptide or epitope per copy, 3 resulting epitopes. Now, if we take epitope availability as a surrogate for presentation likelihood, and the likelihood that at least one of these might be immunogenic, we can see individuals A and B are at an increased likelihood of undergoing immune-mediated cell death. So in this way, this somatic amplification in these individuals with a higher starting epitope burden actually comes at a fitness cost. And we can test this retrospectively. So what we would imagine, if this hypothesis is true, that the tumors that have a high epitope burden that go on to develop somatic amplifications that are immune surveilled should be underrepresented in our retrospective population, as these tumors would have been killed off by the immune system. Okay, at this point, you might be thinking, yes, okay, but these are germline variants. They shouldn't be immunogenic. So at first, we turned to the literature as proof of concept to identify non-somatically mutated peptides that were known to be immunogenic. And we found two, GP2 and E75. Both are 9-amino acid peptides derived from HER2. They are not somatically mutated. So in essence, they're derived from the germline genome. And both have been shown to elicit an immune response in various cancer settings, including going as far as phase 3 clinical trials as possible HER2-positive vaccines. And so we hypothesized that the ability to present either of these peptides, and by that I mean have MHC class I complexes that can bind and present these peptides, would be negatively correlated with developing HER2-positive breast cancer. This is because if you overexpressed or amplified HER2, you would increase the availability of both of these peptides. And if you have MHC complexes that could present them, you'd increase your likelihood of triggering immune surveillance. And so we can test this. We can look at a retrospective cohort of over 600 primary breast cancer tumors, stratify patients based off if they have HLA alleles that can bind and present either GP2 or E75, and then ask if the number of these HLA alleles is associated with whether or not they develop HER2-positive breast cancer. And when we do that, we do indeed see that a high number of these HLA alleles that can present GP2 or E75 is negatively associated with developing HER2-positive breast cancer. So here what I'm showing are patients stratified by if they have a high or a low number of HLA alleles that can present GP2 or E75, and then the ratio of HER2-positive to HER2-negative tumors on the y-axis. And we can see that if you have a high number of these HLA alleles, you're significantly less likely to develop HER2-positive breast cancer. Okay, these are only two peptides. So then we ask, can we systematically look within the full HER2 protein or ERBB2 gene that encodes it? So to do this, we looked at each individual's ERBB2 sequence, including all possible protein coding germline variants, and with their HLA alleles, predicted the number of epitopes that could be derived from their variant germline sequence. We then did the same thing with the reference genome. So this is just the human reference genome, and essentially normalized the number of epitopes that were derived from the individual's germline sequence against the reference genome, and stratified patients based off if they had more, the same, or fewer epitopes derived from their sequence compared to the reference genome. And so this stratification is what we're referring to as the germline epitope burden, or GEB for short, moving forward. And then we can test if GEB is associated with whether or not these individuals developed HER2-positive breast cancer, here considering only individuals of European descent and correcting for 6 genetic principal components, as well as the somatic mutation burden, in order to control for population stratification, as well as other possible confounders like somatic mutations that could also serve as epitopes. And then we can do this systematically across multiple independent cohorts spanning all stages of breast cancer, from pre-invasive ductal carcinoma in situ through primary invasive all the way through to metastatic disease. And when we do that, we see a story very similar to the GP2 and E75, whereby individuals that have a high germline epitope burden are significantly less likely to develop HER2-positive breast cancer. And so here, again, similar to the GP2 E75 bar plot, I've stratified patients based off of their epitope burden along the x-axis, and then plotted the ratio of HER2-positive to HER2-negative tumors in each of these categories along the y-axis. And you can see that the high epitope burden tumors are significantly depleted of HER2-positive breast tumors. Okay, but HER2 is not the only focally amplified oncogene in breast cancer. There are 4 ER-positive subgroups of breast cancer that are defined by focal amplifications at the regions listed here. And so what we can do is something very similar to HER2, and we can calculate the germline epitope burden in each of these representative genes, add that all together, and ask if that aggregate germline epitope burden is associated with whether or not the individual develops the corresponding subtype. And so essentially for each of these subtypes, you can create a bar plot that looks very much like this, and then take the odds ratio here, and I'm plotting now along the x-axis, that odds ratio, and you can see across all of these subtypes, a high epitope burden in the focally amplified genes is negatively associated with focal amplification and development of the characteristic or the subtype that characterizes. And you'll note that here there's only 3 ER-positive subgroups shown. That's because IC6 just had too few missense mutations or protein coding changes in the amplified genes to be sufficiently powered for this analysis. But nevertheless, you see this consistent negative association between a high epitope burden and developing each of these subtypes. And this is our discovery cohort of over 600 primary breast tumors. But we see the same associations when looking at a replication cohort, and this is a meta-analysis of 3 independent cohorts. Okay, at this point, it wasn't clear to us what was actually driving the signal. Was it truly the presentation of these epitopes on the cell surface, or perhaps there was something about these germline variants themselves? Maybe they had some unknown functional impact that hadn't been reported that was leading us to see these negative associations. So to test this, we can basically do the same association as I just shown you. Here in the red box, I'm showing you that negative association between the epitope burden and developing HER2-positive breast cancer. This is the same as what was on the previous forest plots. But then what we can do is we can do this again, but this time hold the germline variance constant and randomly assign the HLA alleles. And why we do this is because if the germline variants themselves are causing this negative association, when we randomly assign the HLA alleles, this should break apart the association, but the germline variant should be held constant. So we should still see this negative association, even with the randomly assigned HLA alleles, if they are not actually what's driving this signal. However, if it's truly the presentation of these epitopes on the cell surface, by randomly assigning the HLA alleles, we're breaking apart our ability to calculate that presentation, and the association should go away. And when we do that, when we randomly assign the HLA alleles and rerun this association, we no longer see this negative effect. In fact, you can see here the predicted odds ratio is 1, essentially null. So this to us suggests that it's not just the germline variants alone, but it's actually the combination of the germline variant in the HLA allele and its ability to present it that's driving this negative association. And this was observed for all of the subtypes, not just HER2-positive breast cancer. Another thing we can do as an additional control is look at sites of heterozygosity. And we can ask if the epitope binding strength at each of the alleles influences which allele is preferentially amplified, almost like an internal control. So at these sites of heterozygosity, you might expect one of 2 scenarios. In scenario 1, the reference allele produces an epitope with a higher binding affinity. If this is the case, in order to avoid immune detection, you could imagine that the tumor may preferentially amplify the alternative allele, the one that has the weaker binding affinity. Or in scenario 2, the opposite might happen, where the alternative allele produces an epitope with a higher binding affinity, at which point the reference allele may be preferentially amplified. And so when we looked at this with considering one common variant within ERBB2, again, the gene that encodes HER2, and we stratified tumors based off if they preferentially amplified the alternative allele or the reference allele, you can see in the tumors that preferentially amplified the reference allele, these tumors, the alternative allele produces an epitope with a higher binding affinity. So here on the y-axis, this is showing essentially the binding affinity differential between the alternative and the reference allele. It can be also thought as the alternative allele binding strength. And so you can see when the reference allele is preferentially amplified, the alternative allele binding strength is higher than when the alternative allele is preferentially amplified. In other words, the allele with the weaker MHC class 1 binding affinity is preferentially amplified. There's the inverse effect. And this is just one common variant, but we see the same thing when looking at a second common variant in TEB-D1. Okay, so this germline-mediated immunoediting, as we've now come to call it, is not complete. A subset of tumors that have a high epitope burden still do go on to develop a somatic amplification. So we wanted to know why that was the case. We reasoned that these tumors might have developed mechanisms of overcoming this immune surveillance. And so to address this question, we turned now to the Cancer Genome Atlas and asked if there was differences in the immune landscape between tumors that had a high germline epitope burden versus tumors that had a low germline epitope burden. So here what I'm showing are HER2-positive tumors. We took 23 transcriptional immune signatures. These were previously published by Thorson et al. As part of the TCGA consortium. And you can see just simple clustering of these tumors identifies two broad categories. Category 1, there's these group of tumors that can be characterized as myeloid predominant. In category 2, there's a group of tumors that can be characterized as lymphocyte predominant. And so we wanted to know if these broad categories correlated with the germline epitope burden of these tumors. And so what we found when we stratified tumors based off of their germline epitope burden, here low versus high on the x-axis, and then whether or not they fell into the myeloid predominant in red versus the lymphocyte predominant in blue, you can see that high epitope burden tumors were actually significantly depleted of that lymphocyte predominant cluster. And the same was true in the ER-positive tumors, suggesting that these high epitope burden tumors that still go on to amplify their burden higher via somatic amplification have developed mechanisms of overcoming this immune surveillance and end up with a lymphocyte depleted tumor. How they do this, we're not sure. We just know that the end result is a lymphocyte depleted tumor. And we can replicate this in an independent cohort, this time looking at the Metabrick cohort with imaging mass cytometry data to identify T-cell infiltration. And you can see in both HER2-positive and ER-positive tumors, a high epitope burden was once again negatively associated with T-cell infiltration in these tumors. Okay, next we wanted to know if the germline epitope burden is prognostic, thinking that perhaps these tumors that have a high epitope burden that go on to develop these somatic amplifications and now are lymphocyte depleted may actually be more aggressive than their low epitope burden counterparts. So this time, once again, we turn to the Metabrick cohort, which has over 2,000 primary breast tumors and pretty comprehensive clinical follow-up. And within each of these subtypes, stratified tumors based off if they had a high epitope burden versus low epitope burden. And we found in HER2-positive tumors, a high epitope burden was indeed associated with decreased overall survival at 5 years compared to the low epitope burden tumors. And the same thing was true in ER-positive tumors, although you can see the effect size is much smaller. But nevertheless, these high epitope burden tumors had decreased overall survival compared to the low epitope burden tumors. And so to further support this and ask if this epitope burden was actually increasing the tumor's proclivity, perhaps proclivity to metastasize, we investigated if there was a difference in this germline epitope burden between the primary tumors and those that actually went on to establish metastatic lesions. And so here we compared the germline epitope burden in two primary cohorts, the TCGA and the International Cancer Genome Consortium, or ICGC, against a series of metastatic tumors from the Hartwig cohort, hypothesizing that perhaps these metastatic tumors would have a higher epitope burden than the primary tumors. And indeed, that's what we saw. So here, this forest plot is showing the association between the epitope burden in metastatic versus primary tumors. And you can see across all of these subtypes, metastatic tumors had a higher epitope burden than primary tumors, suggesting that tumors that are able to overcome this epitope burden may have increased propensity to metastasize. And then finally, to investigate if this increased aggression may be due to the tumor's ability to overcome this immune surveillance, we asked if we could kind of take a step back in time. Here, looking before the establishment of an invasive lesion to the pre-invasive ductal carcinoma in situ stage. And so this DCAS stage, which is here represented by the schematic in the middle, the myoepithelial layer around the duct is still intact. And so we reasoned that at this stage, the tumor may not have developed mechanisms of overcoming this immune surveillance and therefore may still be susceptible to immune detection and immune surveillance, really, and hypothesized that prior to the establishment of the invasive lesion, this germline epitope burden may actually be protective and may decrease your risk of invasive breast cancer recurrence. And this is a small, relatively exploratory cohort, but nevertheless, when we stratified which lesions we knew went on to recur as invasive breast cancer versus the ones that didn't, you can see the DCIS lesions that did have an invasive breast cancer recurrence were actually depleted of high epitope burden tumors, suggesting that at this pre-invasive stage, this epitope burden may actually be protective against invasive breast cancer. And so really, if I could just leave you with a few take-home messages, it's that we see evidence of germline-mediated immunoediting in breast cancer, where a high burden of germline-derived epitopes in an oncogene is associated with reduced likelihood of further amplifying that oncogene, as this only increases the epitopes available and the likelihood of immune surveillance. And at the pre-invasive stage, this high germline epitope burden is protective and associated with a decreased risk of invasive breast cancer. But once that tumor is established as an invasive breast tumor, this prognostic association is inverted. And now this high epitope burden is actually associated with worse prognosis. And we think this is because there's pressure from the immune system to develop mechanisms to suppress or evade this immune surveillance earlier than the low epitope burden tumors. And in this way, this germline-mediated immunoediting may be a mechanism by which our germline genome influences which somatic mutations a tumor acquires, or at least which ones persist to be measurable at the time of a diagnosable lesion. And so, like I mentioned at the start of this talk, this work was really a product of my time in the Curtis lab as a postdoc. And so there's a number of people in the Curtis lab that I would love to thank for this, but particularly Noah Greenwald, Aziz Khan, and Christina Sotomayor. Of course, really like to thank my supervisor at the time, Christina Curtis, as well as our collaborators, Michelangelo and Rob West. And then I have started my own group, as I mentioned, at McMaster University. I'm situated between both the Department of Biochemistry and Biomedical Sciences, as well as the Center for Discovery and Cancer Research, which makes it a really fun interdisciplinary group. We have clinicians, we have virologists, we have immunologists, we have statisticians, we have really the gambit here. And we are hiring both grad students and postdocs. So if this is of any interest, please feel free to email me, and my email's on the slide. And thank you so much all for listening.
Thank you so much, Katie. That was a very, very great story. And I think I'll let the audience digest a little bit. If you have questions, feel free to either type in the chat or raise your hand. I think we have few enough people you can directly ask. I like to start with some so intriguing, Katie. So I think especially immunology, right? But we are taking the genomics approach to it. A lot of the negative results were never revealed. And I bet it must not be easy to have done this. And what are like the negative results that didn't go in? that you think are intriguing?
Yeah, it's a great question. So, one thing that we touch on a little bit in the paper, but I didn't include it here, is that we really, interestingly enough, don't really see the same effect in triple negative breast cancer, at least not to the way, um, not to the extent that we see it in this HER2 positive and ER positive setting. And I think that could be a consequence of a number of different things,
Mhm.
including perhaps the higher somatic mutation, like point mutation burden in triple negative breast cancer compared to ER positive and HER2 breast cancer. It also could be due to the fact that in both HER2 positive as well as, I should be clear, I'm not talking about all ER positive breast cancer. I'm talking about a smaller subset that has these focal amplifications.
Mhm.
But at least in these tumors, we do see really clear focal amplifications that are, the characteristic amplification for that tumor, which you just don't see in triple negative breast cancer. You see more broad, diffuse genomic instability. And so, you know, I do think that there are limits. Well, I do believe that this mechanism is probably more generalizable than breast cancer. I think that it's going to be a very specific uh, either tissue type that maybe is slower growing that can have this sort of back and forth interactions with a very specific copy number profile and a limited number of somatic mutations that might be out competing these germline variants for presentation and immunogenicity. So yeah, I think it's important to think of this in the context, and I do think it's generalizable, but I don't expect it to be generalizable to melanoma, for example.
Right. Yeah, because that's directly related to my colleagues here, Robbie Simstein's and Diago Chowell's lab. They were showing that MHC heterozygosity has to do with cancer risk. And I think what's actually surprising in that paper is that, I mean, they obviously reported lung is where the effect is, but then almost all the other cancer types didn't have the effect. And okay,
Yeah.
then how that relates to yours also, like, I don't know if you have checked this because it will occur to me that these high GEB tumors may have likely invaded immune system through loss of HLA, but I feel like if it's just that you have checked.
Yeah, it's a good question. So first off, yeah, I think their work on HLA, I believe it was predominantly class two, right? Heterozygosity and lung risk. I think it's fascinating, and I think it might have something to do a little bit with the smoking status of some of these tumors, but I think it's really supportive of some of these approaches. I think we're kind of looking in breast cancer, we don't see HLA as they saw, we don't see HLA heterozygosity alone is sufficient to describe this signal. It's the HLA with the germline variant together is how we see this effect.
Mhm.
But it is really interesting that they see it in lung, and it's something that I would like to follow up with the GEB to see if we see something similar. To your question about HLA loss, yes. So I looked at HLA presentation through a variety of transcriptomic signatures. And while we did see that high epitope burden was associated modestly with reduced HLA presentation, it wasn't strong enough to definitively say, this is the mechanism. So I think it's playing a part, but I actually think it's probably a number of different mechanisms
Mhm.
that are playing a role on this, not just HLA presentation or HLA LOH on its own. Though, of course, I think it's a mechanism.
Yeah, that makes sense because I guess the hard thing about this, you're subgrouping and a subgrouping of a subgroup, then you lose more and more statistical power. Yeah.
Yeah. And these are weak effects for sure. Like I think when you talk about germline variants just in general, we're talking about each individual variant having a very weak effect that we're only seeing this signal when we aggregate these variants up across, a number of different genes using a really big population size. So yeah, I just kind of want to contextualize that these effects are robust, but they're not really strong effects, more similar to the GWAS studies that you see.
Right. Okay. Um, Everyone else, I guess it is a little challenging to grasp. I think the paper because you require understanding of genomics and cancer and immunity at the same time. But everyone else, please think, if you have questions. The other key question, Katie, is
Yep.
diversity, right? As you show, like,
Yeah.
like the diverse genome has much more different variation
Mm-hmm.
compared to the reference. And so, and obviously the HOA is even more different, right?
Mm-hmm.
So do you include all that and how do you navigate that with like the real signals?
Yeah, it's a great question. So unfortunately, in order to reduce confounding from, different HLA frequencies, different variant frequencies across different ancestry populations, we've had to be really focused and just consider individuals of European ancestry. And we further correct for genetic principal components to kind of correct for more finer structure, population structure. So all of this work has been just focused on individuals of European ancestry, as that's where we have the majority of the data thus far. But I really look forward to exploring these in other ancestry populations, and I think this could help explain some of the differences in frequencies of different, subtypes or somatic variants across ancestry populations. So yeah, that's absolutely a next step. But for this analysis, it's been unfortunately more focused just to ensure that we weren't confounded by differences across ancestry populations.
Right. Do you think the GEB will have a cancer risk effect?
Yeah, that's something we're actively exploring. I
Okay,
think I think it
great.
very well could. Yeah.
Yeah,
And
that's
and may
awesome.
have been missed from previous GWAS studies because it really takes both the variant and the HLA allele in kind of an epistatic way as opposed to the individual variants.
Yeah, yeah. I guess the hard thing there is that, late in infancy HOA psychosity on risk, right? Maybe the signal is just it will require much careful teasing out probably. Right.
Yeah, it's going
Yeah.
to be thoughtful about which genes you consider. It's going to be thoughtful about
Yeah.
how you define epitopes. Yeah, it's non-trivial to think through these. So it's going to have to be a lot of sensitivity analyses to make sure our choices are not biasing our results. But yeah, it'll take some careful thought for sure.
Right. Okay. Anyone has any questions? Even if you need a clarifying question, feel free to ask because if you have a question, some other people might have as well, or have comments, thoughts, and take a minute to wait. Maybe I guess the technical clarifying question that may help some audience, Katie. So when you calculate the GEB, you are taking each individual specific HOA and their germline variants. Do you take both their copy of HOA? Do you take one and then to make sure that's the expressed one?
Yeah, so it's a great question. So we're not going as far as looking at the expression of the HLAs. We're really just taking the average across. So it's quite literally the mean of the number of epitopes across all six HLAs. So, HLA1A, HLA-B, HLA-C,
Mm.
and then both copies. And then taking the mean of the number of epitopes predicted from each of those.
I see. Okay, so the HOA you are using is not personalized yet at this.
It's personalized to that individual. So we're taking their genotype of like their
Oh,
copy of HLA-A, their copy of
I see.
HLA-B, both copies, HLA-C, both copies. So each individual will have six different HLA alleles,
Mhm.
and then we run the prediction and then take the average across all six.
I see. I see.
Yeah.
Is there a better approach than taking the average?
Yeah, it's a great question. I mean, we've been I will say I suspect our epitope predictions are at best about 60% accurate. So in part, we are really binning the signal. And you can
Mm,
see here we bin it by greater than the same. Like we're kind of trying to take the aggregate as much as possible, just to see if we see signal, because we don't anticipate our epitope predictions are super robust. So we're working on refining those. But for here, it was more if we aggregate this signal broadly or the measurements broadly, can we pull out signal as proof of concept? So yes, I absolutely believe that there's better approaches, and we're working on implementing those now. But
mm.
this was more just about getting a kind of burden aggregate at first to see if we could see signal.
Mhm. Okay, I think we'll wait for the last 1 minute if other people have questions, and I'll ask a dumb question in the meantime. Like, so with all the AI breakthrough, is there an AI breakthrough in because I know, epitope prediction. When I was at Wall Street, people were coming up with first software, and everyone is like, "Oh, this field we never know which is a real signal." What's the current state? Is there like a big breakthrough thanks to all these AI or not yet?
So it's coming. I wouldn't say it's the problem. I don't think is like the models.
Hmm.
I think we have models that we can use to predict. The problem is the training data. So the problem is more these like really high throughput, robust methods of detecting immunogenicity or detecting presentation. And so there have been a few. I mean, Steve Elridge has a couple. Polyfordice has one. And so data is being generated from that. And you still see papers where people are pulling from like IEDB, which is, a big database of immune different types of assays for T cells, B cells, et cetera, to measure immunogenicity. But I think part of the concern is just like the batch effects and the how systematic those are. So it's changing. I think with the increase in these assays that can do this in a high throughput, we're going to get a lot more data, which will help us build out models. But yeah, I don't think the rate limiting step is the model. I think the rate limiting step is the training data right now.
I see. I see. Okay, cool. Then I think Katie, that is so helpful, and that's such an interesting story. I'm so happy we did this because I think it will help a lot of people clarify it in the paper, and having it available on YouTube will really help. So thanks so much, Katie, and thanks
Great.
everyone.
Thank you. Thanks all.
Bye.